start by connecting what we're talking about. So we ended up talking about action potentials on the last day of class, last Wednesday, right? Before that, we were also talking about the cytoskeleton. And the last thing we talked about with respect to the cytoskeleton was the actin and myosin contractions that we have in muscle cells. So what's the connection between the two? Well, we're talking about action potentials in nerve cells that are going to signal for the contraction of these actin-myosin fibers. So the nerve cells are going to send an electrochemical signal that is going to be turned into a purely chemical signal, which is going to signal to those muscle fibers. And remember, by muscle fibers, we're referring to essentially a bunch of cells in the same membrane, because we don't have that final step of actually separating the membranes after mitosis in muscle cells. And these muscle fibers are going to receive the signal from nerve cells, and then this is going to cause the myosin and actin to contract and shorten the muscle. And we'll talk more about the molecular basis for the end of that process in a little bit. But first, I want to finish our discussion of action potentials. So we talked about some of the molecules that are important for this process, and I drew them on the board here. So I think that we talked about the sodium-potassium pump. So does anybody remember anything about the sodium-potassium pump? So for example, how does it work? Is it free or does it cost energy? Yep?
It costs energy and it controls filtration in and outside of the cell with ions.
Right, so it uses ATP, so I'm going to draw ATP over here. And I just drew it in the same shape that you'll see it in your textbook, so that's why it's drawn kind of funny on the bottom here. So it binds ATP and it hydrolyzes that to ADP, and it's going to pump out our sodium, and it pumps in potassium. And as we were saying, it's actually going to pump out 3 positive charges and pump in 2 positive charges. So our net is 1 positive charge out. So we might say that the sodium-potassium pump is making the inside of the cell more negative, but I think a more accurate way to say it would be to say that it's making the inside of the cell less positive, because it's only moving positive ions, but it is removing 3, and bringing in 2. So we have more negative the inside of the cell. And also, if you recall, in the slides we saw before and in your textbook, it depicted proteins with a negative charge near the membrane. Does anybody remember one of the reasons for these negative charges on proteins? I think we discussed 2 things. What's one chemical group that we've talked about adding onto proteins?
Phosphate groups?
Phosphate groups, yes, and those have a negative charge. And then what's one other thing on a protein that might have a negative charge? On only perhaps certain proteins with certain compositions? Anybody remember the first question on the first exam? No. You say negatively charged amino acids?
It was about the class of amino acids.
Yes. Yes. The classes of amino acids was the first question on the first exam. And some amino acids have a functional group that have a negative charge. So that could also be providing negative charges inside of the membrane of the cell. Okay, so at rest, the cell is negatively charged. The outside of the cell is positively charged. What are we looking at right here? Well, we're actually looking at this part of a neuron. We're going to be focusing on this part right now. So basically this long extension of the cell, which doesn't have very much in it. It's mainly just cytoplasm and membrane and all these specialized membrane proteins, transmembrane proteins. And so this is the axon. This is where the action potential is going to be propagated. So when the action potential or the signal comes for an action potential, it is actually sent by the main cell body. So the cell body, and we'll talk a little bit about this in a couple of minutes. The main cell body is initially going to send the signal to the axon that an action potential needs to be propagated. And this signal will be in the form of a change in charge, which is the form that the action potential takes as it moves down the axon. And it's going to be a net positive. So the cell is going to become more positive inside than negative. Okay, so how does this occur? In addition to these sodium-potassium pumps, I think we might have also talked about these voltage-gated pumps. Did we talk about these on Wednesday? A little bit? Okay. So these guys, I drew this one open actually. But at rest, when an action potential is not going down the axon, these are closed. So these are voltage-gated sodium channels. And they're called voltage-gated because they, instead of responding to a chemical signal, which is what we've talked about proteins responding to in the past, these are going to respond to a change in charge. So when we get the positive charge coming into the axon, these voltage-gated channels, I'll write that up here, will sense that change in the electric charge, and they're going to open. Okay, so these guys are channels. They don't bind to ions and use ATP to bring them across the membrane like the pumps do, but they're still specific to only letting sodium flow through them. So when they open, we're going to have all this sodium coming in. Why is sodium coming in? Yep.
Because of the gradient, because it wants to be in more negative.
Right, so we set up a gradient with the sodium-potassium pump. We've been pumping it out all this time. Now it's flowing in down its concentration gradient in terms of the chemical gradient, and it's also flowing down the gradient in terms of the electrical gradient, because remember, the cell was more negative on the inside. Okay, so we have the opening of this channel, and then all these positively charged ions move in, and they're going to move away from the channel. So part of the reason why the action potential is going to move down the axon, so it's going to move down the axon here, is because these cations are going to interact with things that are already in the cell. So what happens when you get a bunch of positively charged molecules together? Do they attract or do they repel? They repel. So if there are any positively charged ions over here already, they're going to be pushed forward down the axon. And so that's going to start the opening of these voltage-gated channels, and then you're going to get even more sodium rushing in. Okay, so how do we recover from this? Why don't we just have the whole cell fill up with sodium permanently? Well, this voltage-gated sodium channel has another subunit. So that's what I've drawn here. And this subunit actually changes conformation to move to block the channel. So you could refer to this as a stopper or just a second subunit. And this is time dependent. Last week we talked about microtubules and how GTP is hydrolyzed to GDP, right? But we also talked about the fact that this happens slowly. So this is a slow reaction. You could think of this in the same way. So when this channel opens, that's a conformational change. And when that happens, it stimulates the next conformational change, which is going to be the adding of the stopper, but that part happens slowly. So you have the opening of this, which is essentially pulling the stopper to block the channel, but the stopper moves into place very slowly so that you get lots of sodium coming in before the stopper goes into place. Is that clear? So here's another question. In general, why might we get a conformational change in response to a change in charge in the first place? What are proteins made up of?
Amino acids.
Kittens? We wish. Okay, amino acids. And we know that amino acids can change conformation with respect to one another in a protein. And we've talked about how this happens when it binds another chemical, but this can also happen when there's a change in the environment, like pH or the electric charge. And so we won't go too deep into it, but essentially you could think of that as the reason why we have this change. So again, the charge is causing the interactions between the amino acids to change so that you have this conformational change. Okay. So now at this point, we have had lots of sodium come in. We're not having sodium come in anymore, at least in this area of the axon. But we need to restore the negative charge on the inside of the cell if another action potential is ever going to come through, right? Because otherwise, you would never be able to depolarize the membrane again. So does anybody have any predictions as to how this happens? Using one molecule we've already talked about. Yeah.
Do you pump potassium out after you do the channel?
So, not directly, but we continue to use the sodium-potassium pump. And so we're pumping the sodium out. And in addition, we have these other transmembrane proteins at work. So we have one channel, which is called the leak channel. And this guy is actually always open, or rather, it's always moving between an open and closed configuration. So this is, again, this is a potassium channel, But it moves potassium out of the cell slowly because it open, close, open, close. So it's not like this very, very fast, large effect that we have with the sodium channel. In addition, we have a voltage-gated potassium channel. And this channel also has kind of a time-dependent effect. So these voltage-gated potassium channels open or begin to open when you get the inside of the cell starting to become less negative. But they don't open immediately. They're slow pokes. So as all this sodium is coming in, you have the opening of this channel, which again is slow. But we have a conformational change that's going to cause it to open.
Wait, it changes when it becomes more negative or less?
Less negative. Apologies. So as the cell is becoming less negative, because the sodium is rushing in, these guys begin to open. They open slowly, so they're only fully open when these guys go into their closed conformation. So again, this is the slow reaction. This is also slow reaction. So they happen essentially more or less at the same time. Yep.
Is that a channel or a pump?
So this is a channel. Both of the voltage-gated things are channels. So here we're having the potassium move along its gradient because we have the inside of the cell, which is more positive at this point. So we're moving positive molecules along the gradient. And we had also pumped all the potassium into the cell here. So there's a higher concentration of potassium inside the cell, and it's moving out. Okay. So eventually, the cell is going to recover. This voltage-gated channel, it will remove the stopper, but it's going to be closed still because it's not going to use the right voltage. This. The stopper will be just hanging out. And then these voltage-gated channels will be closed as well. And then we'll continue to just have the sodium-potassium pump. So we're pumping out the sodium, which is going to be important for when we need to open these and have sodium rush in. And in addition, we're building up the potassium on the inside of the cell. And potassium is leaking out at the same time, which is just allowing this cycle to continue. Yeah?
What makes the voltage graded ones like close?
What makes them close?
Yeah.
So this one, the sodium one, the stopper goes into place over time. And then once it's stoppered, then you have the recovery of the cell to the more negative state. And so it won't be opened due to voltage-related reasons anymore.
Okay.
Yeah.
Cool. Thank you.
And same thing with the potassium one at the end there. Any other questions? We're good? Kind of good? Okay. So, we've talked about that potential moving down this part of the membrane. Have you guys ever seen the depiction of an action potential on a graph showing the charge inside of the cell over time? Yes? A couple people? Okay, so that is actually in your packets. I'm going to ask you to take 2 or 3 minutes to actually fill that out with the person next to you and to label what's going on with these different transmembrane proteins that causes the changes in voltage that you see.
I think we did, it has like numbers, I think we just say 1, 2, 3. As the cell becomes less negative,
And then
the potassium channel is closing.
Is that just like the start? Like that because it should start opening and I can start it?
Yeah.
I feel like it's the potassium channels opening to like basically like the positive charge. So you are
Yeah, I agree. I didn't get that.
I don't know. I think
I
that's
think it
actually
just
very
like
fast.
closing and then like returning back
So
to
once it after isn't that what they go to start?
like then it goes straight to
I just say on the way down, it's like hit the threshold potential, like signal to stop, and then it goes to like to return to the rest.
There is a ton of ink left in this paper. Why is it not working?
Well, the threshold would be like what signals it to like start the action potential. Stop and return to the rest. Here's the question.
You could try. I don't know anything about this.
Okay.
I study meat, which don't have neurons.
Radius is that like returning back to
how I was like, this is negative and this is positive. This is positive, this is negative. The opposite of what I thought it would be. In the sense this is hyperpolar.
Okay, so we know the resting potential is negative for the sodium potassium bonds, which is keeping it negative. And then depolarization happens because the voltage gated sodium channel opens and then lets in all the positive ions. And then it repolarizes when that closes and then the voltage gated potassium channel opens and all the positive charge flows back out. Yes. Okay. I hate self-scoring.
Okay, so once action, what's going on when we're at the resting potential, so the beginning here, so before there's any action potential? What's working? Yep. Everything voltage gated is closed. What is open? Two things. Yep. The leak channels are opening and closing. They are always doing this. And then what else is active? The pump. The sodium potassium pump is also active. So that is making the cell more and more negative, but the cell doesn't get super negative because at the same time you have those leak channels letting out some of the positive charge in the form of potassium. Okay, so next step. Okay. Depolarization. So this is when we get that signal and what happens. I shouldn't have this up here if I'm asking you guys questions. Pretend you didn't see it. Okay, so what happens? Yep.
The voltage-gated sodium channel opens and the cations flow into the cell.
Right. So we have lots and lots of cations, positively charged sodium ions flowing into the cell because we have this positive charge coming through, which stimulates the opening of these guys. What else do we still have going at the same time that's always going? The leak channels and the pump. You're the pump person now. Okay, so the sodium-potassium pump and the potassium leak channels are always going no matter what. They don't make that much of an effect on the overall charge of this region of the cell at this point because the sodium channels opening just let in so many cations at once. Okay? And so that's why the cell becomes neutral here, and then it actually has a positive charge for a bit of time. So why do we not continue getting more and more positive? What happens at the peak there?
The voltage gated sodium plus channel closes.
Right, so we have the time-dependent closing of the sodium channels with the stopper. And then what else is going on at this point? Yeah.
The voltage-gated potassium channel opens at the same time.
Right. The voltage-gated potassium channel opens. This process began around here, but it takes them so long to open that they don't fully open until we have the sodium channels closing with the stopper. And then again, the leak channels and the pump are still going. So now we have the potassium going out through both the leak channels and the voltage-gated channels. We don't have any more sodium coming in, so we're having a net loss of positive charges at this point. And in addition, we also have the pump going. And with all three of these things at once, you get an overshoot. So that's why we see this little extra negative region here when the cell is more negative than it usually is. And does anyone know the significance of this physiologically for the cell as a whole? Do we think that a cell could propagate an action potential? Who wants to say yes? Who wants to say no? Who doesn't want to vote? You guys didn't even vote? You didn't want to vote?
What's the question again?
So, do we think that when the cell is this negative, is it going to be able to propagate an action potential? So it's probably not going to be able to. It would be a lot harder to bring up the electric potential to the point where you're going to actually open those sodium voltage-gated channels. So this is it's called an undershoot or the refractory period. And then finally we get back to the starting point where we just have the sodium-potassium pump and the leak channels. And at this point, we're making the cell more negative by using the pump, but we're kind of keeping it in balance by using the leak channels as well, which work more slowly than the pump, but it's making sure that the cell is not becoming super negative. Yep?
So what's the benefit of it not being able to propagate another signal? Why do you want it to not be able to do that?
Good question. So part of it is to make sure that the cell has recovered fully. And all of the proteins that are time dependent have had time to fully go back to their original conformation. And it also means that you would need a very strong signal to respond at this point. Any other questions about this? All right, so let's go in. And of course, you can see all this online. So there's a second set of slides that will be available at noon that have all these details on them, because I know that the handouts and the slides right now do not, because I wanted you guys to work through this. Okay, so getting back to the big picture, when we're talking about how is this going to cause muscles to contract, well, we have action potentials going down the axons of our nerve cells. We have presynaptic neurons and postsynaptic neurons. In our case, we can also have, so we could have things, this cell in the nervous system, and then this cell might be connected to the skeletal muscle system. So we have the action potential moving down the cell, and then at the end of the cell, the little foot-like thing here, which will be at the end of the cell, we can't pass on the action potential anymore because there's no more cell membrane, but instead, the positive charge on the inside of the cell stimulates these channels at the end of the cell. And these are voltage-gated calcium channels. And the voltage-gated calcium channels let calcium come in, and calcium is often used as a signaling molecule in various types of cells. And so in this type of cell, for example, it's going to cause the fusion of these vesicles with the cell membrane. So what are two proteins that we talked about that will allow these vesicles to fuse with the appropriate membrane? Yep.
The snare and rab proteins?
Right. The snare proteins and the rab proteins are going to be on the vesicle and the target membrane. They're going to interact and pull the vesicle into the target membrane so that it fuses. And then what was in the vesicle is dumped out into the space. It's a very small space in between these two cells. And in the case that we're talking about, what's in this vesicle? It is a neurotransmitter called acetylcholine. Who's heard of acetylcholine, at least in passing? Yeah, we've all heard of it in passing. So this is a signal that will bind to receptors on the postsynaptic cell. So in muscle cells, for example, we have acetylcholine receptors. So this is the receptor. I feel like this image is a little bit overcomplicated. You don't need to know how many subunits it has. All you need to know is that there are binding sites for acetylcholine, and that this receptor is also a channel. And the channel will open when it's bound to acetylcholine. So again, when this cell receives the acetylcholine that was released from vesicles, then it will bind it and it will open. And similar to what we just talked about, we're letting positive ions into the cell. So this is again going to cause a signal that is an electrical signal. And this is going to cause, this, in particular in muscle cells, this positive signal stimulates this compartment called the sarcoplasmic reticulum, not the endoplasmic, but the sarcoplasmic reticulum, to open certain channels in its membrane. So we haven't talked about this before. Specialized for muscle cells is just a membrane-bound compartment that has lots of calcium in it. And the channels to release this calcium are stimulated by the signal of acetylcholine binding and letting in these cations. And so then the calcium is released from the inside of the cell. So this compartment called the sarcoplasmic reticulum releases calcium. And all this calcium interacts with the actin-myosin fibers and allows the myosin to initially bind to the actin. So before this, the myosin was blocked from binding to the actin, so you can't get contraction if myosin's not bound to actin, even if you do have ATP around. But when you have calcium, then myosin can actually bind the actin, and then if you additionally have ATP, then you could have contraction of the muscle fibers. These fibers sliding past one another. So in your textbook, it goes through and talks about the particular proteins that are blocking myosin binding and how the process occurs with myosin binding at a certain point. All I want you guys to know is that calcium allows myosin to bind, and that you have to use ATP in order to bind myosin to actin and pull the fibers so that they contract. Are there questions about this? What does this kind of shed some light on in terms of our real lives? So why might you get very hungry after you go to the gym? Why are you tired? Yeah.
Because you've used up a lot of ATP through muscle contraction, so you need to replenish by eating.
Right. So every time you have muscle fibers contracting, you're using lots of ATP. So it's not shown here, but I think it was in our slideshow last week that we saw that there are lots of myosin and actin interactions along a single muscle fiber, and all of them are using ATP, and you're hydrolyzing that ATP every time the muscle fiber contracts. So when you're using lots and lots of those, you're hydrolyzing lots of ATP, and you're using up lots of energy. Yeah?
Just with the vesicles, what did the calcium do?
Can you repeat that?
With the vesicles?
Oh, with the vesicles, they released calcium from the inside of the cell. So you're talking about when we're already looking at the muscle cell?
No, when we're looking at in the cell at the body.
Like so. Let's see.
Yeah, there.
Okay. So this is not in the muscle cell. This is in a nerve cell. The calcium here is going to allow the vesicles to meld with the membrane and release the neurotransmitter. So the calcium in this case is helping whatever protein, perhaps one of the snare proteins or the rad proteins, go into the correct conformation so that the vesicle could bind with the membrane. Yep.
And then how do you get from the acetylcholine binding to the channel to the muscle cell? What was the link between those?
All right, and so there we have acetylcholine binding. This opens the channel and allows cations to move into the cell.
Muscle cell or
The
another
muscle
nerve?
cell.
Oh, okay.
Yep.
Why not have not have an axon? Why does it have to travel that far, and why can't you just have an action potential just in the main cell body connected to the club foot, and why does it have to travel?
That's a good question. And this structure helps the action potential travel the fastest. Because if you think about it, our nerve cells are quite long. We need communication between our brains and all of our muscles, etc. And you need signal for sensing whatever environmental cues, and then a signal back to respond to them. And so the action potential travels faster than a chemical reaction, which we're going to talk about, I guess, next week. But on that note, we can't just have the cell body because we wouldn't have something thin enough that would be able to propagate the action potential as well as the skin.
Yeah. You mentioned the sarcoplasmic reticulum. So when the receptor opens, that allows like positive ions to flow in, and that leads to calcium being released from the sarcoplasmic reticulum. How?
So there are voltage gated channels on the sarcoplasmic reticulum.
Oh, okay.
Yes,
Yeah.
so those are calcium channels that are open when you get this positive charge. And then calcium sort of goes into the cell from an inner cell compartment. Good? Any other questions? Okay, so we could end there, I think. No homework this week. Happy Thanksgiving. I am having students over for Thanksgiving. As you may know, some professors have students over. If you don't have somewhere to spend Thanksgiving, I have room for 1 or 2 people if you like baked turkey burgers. They're really good.
Thank you.